Hello, everybody, and welcome to Connecticut River Conservancy's live stream series, Season 2, Episode 6, Endangered Shortnose Sturgeon in the Connecticut River. My name is Stacy Leonard, and I'm the events organizer here at CRC. We're thrilled to have you all joining us today as we welcome Micah Kieffer, our special guest presenter from the Conte Anadromous Fish Research Lab, who will be talking with us about the mysterious shortnose sturgeon. I'll share more about Micah and the sturgeons in a moment. As you may know, LiveStream is our virtual venue for bringing our work and our rivers to you wherever you may be. We'll have just one more episode in this season on June 16th, Indigenous Relationships with Water, led by Justin Beatty. And then we'll put our live streams on hold as we return to in-person events. Hooray! You can view recordings of past episodes on our live stream webpage and find all of our upcoming events, whether in person or virtual, on our events calendar. And I'll share the links to all of that in an email after this presentation. And that will go to all registrants, even if they weren't able to make it today. A few quick details before we move on to the conversation. We encourage your questions, and I know you'll have a lot of them, and we ask you to use the chat box. We'll do our best to answer as many as possible at the end of the presentation. So we're going to just hold them all until the end. And we are recording this presentation for later access, and we post it on our live stream page as well as on our YouTube channel. And again, I'll be sharing all of that in a follow-up email. So now for today's presentation. Sturgeons are among the oldest of living fishes, some estimates dating them back 200 million years. And the Connecticut River is home to one of the few remaining populations of the species. Fun fact that I recently learned, instead of scales, they have bony plates known as scutes, and it makes them look like they're covered in armor. While sturgeon was a primary food source for thousands of years, today this shortnose sturgeon is listed as endangered. Here to tell us about both the stressors facing these ancient fish and the protective efforts underway is Micah Kieffer. Micah is a research fishery biologist from the Conte Anadromous Fish Research Laboratory located in Turners Falls, Massachusetts. It's the Biological Resources Division of the U.S. Geological Survey. So without further ado, I'd like to turn it over to Micah to lead us on. Thanks again.
Very well. Thank you for that introduction, Stacy. As was mentioned, my name is Micah Kieffer. I've been working at the Conte Fish Lab since 1991, and before that, I have done my master's work on sturgeons in the Merrimack River from University of Massachusetts. So today I'm going to focus on the endangered shortnose sturgeon in the Connecticut River, and this photograph here is a nice shot from an airplane. This was before the Conte lab was constructed, right in this patch of open ground right here. But this pictures the Turners Falls Dam way up in the upper screen, the natural river bed, the power canal, and the Cabot Station power generating station. And I will spend some detail on this effort, but I'll be covering other issues as well. So for those of you tuning in, if you had, I gave a similar webinar just a week or so ago on the Merrimack River. So some of what you see today, if you tuned into that other presentation, will be a repeat for you. I'm also steering away from really hard science, so I'm not going to throw a lot of figures at you. It's mostly photographs, but they generally are pretty well received. And I'm also going to be speaking more about the things that I'm involved with at the Conte lab, but there is a host of things that are occurring with sturgeons in this river. So I was going to, or sometimes I try to start off with a humorous anecdote to break the ice, but I will spare everybody that and start off with a current event. Now, this is a photograph taken by researchers on the Detroit River, part of the Great Lakes system, and this is a lake sturgeon that was captured, and it is by lake sturgeon standards perhaps the largest on record. Now, as far as sturgeons go, it's a moderately sized sturgeon, but this is enormous for a lake sturgeon. And what I want to point out about this particular fish is, my eye says this is a late stage female. You can see the width of this fish is very wide, and it's a successfully reproducing female, which is amongst a population. These are amongst the most important individuals because they have the experience and the good genetics and the success of carrying the species through. And so that idea will come up a little bit later. So just a little bit note about sturgeons. They are a primitive fish, and they have a primitive body design. This may look familiar because it's very similar in the design of a shark's body. And therefore, these fish are capable of long and sustained swimming distances. They are covered with 5 rows of these bony plates called scutes that Stacy mentioned, and they are kind of a leftover from the ancient fishes that had much more bony armor on the outside of their bodies. One of the unique elements of sturgeons are what we'll call a protrusible mouth, and not only is their mouth oriented for feeding on the bottom, but they can protrude this muscular tube with its functioning bones, and it can actually dig or jet water into the substrate to dislodge prey species. Sturgeon is what we like to call a living fossil, which simply means that there are species with similar characteristics current in the fossil record. In the sturgeon's case, it's believed they existed as far back as the Triassic period, 200 million years ago, which means that, this fish hasn't changed in an awful long time, which means that it is a very successful animal design. Because it's been on Earth this long, it means it has survived a number of mass extinction events. So it is a survivor, and it has been successful for a long time. And although the images on the right, or on the left, are sturgeon-like fossils, there was recently a discovery of much more descriptive sturgeon fossils found in a 66-million-year-old deposit that was populated with paddlefish and numerous sturgeons. And you can see the unique tail structure and also the pectoral fin and pectoral girdle indicative of sturgeons. So a successful animal by many standards. About 26 to 29 species of sturgeons, depending on who you ask, and they are confined in roughly this band of latitudes. No sturgeons in the southern hemisphere. In North America, there are 9 recognized sturgeon species, and this very ornate poster of North American sturgeon species shows the short-nosed sturgeon here, but it misrepresents the relative sizes of these different species. For instance, the smallest sturgeon, the shovel nose, adults reach about 2 feet in length, whereas white sturgeons have been recorded to achieve 20 feet of size. So there's quite a distinct or astounding range of sizes that these species have adopted. Sturgeons are known as a diadromous fish. Diadromy is a term that means the fish exhibits some form of migration during its, living its life. Anadromy describes an animal that lives in saltwater and spawns in freshwater, like a salmon. Catadromy defines a species that lives in freshwater but goes to sea to spawn, like an American eel. Then there's potamodromy, which is a fish that lives its entire life in freshwater but still makes a spawning migration. For instance, a northern pike that might live in a lake but enter a stream to access spawning sites. And amphidromy is a movement between habitats that is unrelated to spawning, like a striped bass pushing upstream for forage. So just a bit on global decline of the sturgeon species. In about the mid-1800s, commercial harvests of sturgeons crashed. And in hindsight, these the issues that are attributed to these crashes are, we'll start with the migration obstruction. Now, this is a spillway of a dam. This is in a Midwestern river. And it's hard to see in this photo, but there are a few dozen small black triangles at the surface of the water. These are all lake sturgeon tails gathered in a shallow water. And the assumption is that they're trying to push upstream and get beyond this obstruction. Overharvest was another big issue. Sturgeons are slow to mature and infrequent spawners. So an overharvest of, again, these valuable late stage females can be catastrophic to the population, recruiting new members. Drop down here to water quality. This is just an example of a fish kill, but ask anybody that's lived in New England for a long time and they can tell you about the stories of knowing what color shoes they were making today because of the color of the river and the dyes that have been dumped into the rivers. And I have here water flow alteration. This is an example of a site in close proximity to a known sturgeon spawning site. And what can happen in a situation like this is an emergency spillway. And a female sturgeon who makes all the decisions about where spawning happens may choose this particular spot as the perfect spawning site, lay the eggs and leave. And then hours or days later, the flow is significantly altered into a condition that a female would not have selected. So we've made quite a lot of progress on some of these items, particularly water quality. There's still issues with flow regulation and obstructions. And at least in this country, most of the populations are not harvested. Part of the decline of the sturgeons were, of course, they were captured for meat, but the big money maker, of course, was the caviar fishery. And it'd be nice to think that the eggs from a late stage female could be extracted without killing the female to gather the eggs. But by the time they're flowing out of the female naturally, they're overhydrated and unsuitable for caviar production. So to make caviar, a female has to be killed. So currently, there are only 14 remaining reproducing short-nosed populations left on Earth. And this was typically the entire range of short-nosed sturgeon here along the eastern seaboard of the United States. So I'm going to be talking about the Connecticut River here in the mid-Atlantic area. So how do scientists go about gathering these types of information? In order to capture fish, we use a millennia-old technique of setting gill nets because of sturgeon's physiology and behavioral demeanor. They get caught in a net and they don't seem to exhaust themselves trying to escape like a salmon or shad would. So they appear to take a more docile behavior once in the nets, allowing this to be a viable capture method. Using a little bit more advanced technology, these are telemetry transmitters that can be attached externally or internally to sturgeons, allowing us to get a much more intimate look at their movement patterns. Now, suppose you had a good day of fishing on your research vessel and you capture 10 sturgeons, but you only have 5 tags. How do you decide which fish to tag? We've developed a a technique of using a fiber optic borescope, which you see inserted into the genital canal, genital opening of a sturgeon. And by using, we learned through dissection of a recovered species mortality that the duct through which eggs travel is translucent. So we can actually go inside without doing any invasive procedure. When I say invasive, I mean surgery, and tell if there are eggs inside the fish and even what stage of development those eggs are. So down here in the lower left are early stage eggs. They're smaller and lighter and will not be ready for spawning for the next several years, perhaps. But the fish on the eggs on the right are the larger late stage eggs. So the female is physiologically ready to spawn the next spawning season. So if your study objectives are to, let's learn where the spawning is occurring, tagging this fish would be far more valuable than tagging this fish. So without causing too much discomfort to the fish, we're able to have a look inside to help us decide which fish to pursue. And we also, during the course of my career at the Conte lab, have adopted, you can learn a lot from telemetry, but unless you capture viable eggs or embryos, early life stages, it's difficult to really say that we know this is a viable spawning site. So we use nothing more complicated than a modified plankton net set downstream of where tracked fish tell us is a likely spawning site. And the eggs in the upper left are showing advanced cell division. You can actually see structures forming. So these were viable fertilized eggs, in other words, successfully spawned eggs. After about a week to 10 days, these eggs hatch into a free embryo, large yolk sac that the fish needs to feed off of for the next week to 10 days. You'll notice there's no sensory organs and there's really no swimming ability of this fish. So what it basically does is it finds cracks and crevices in the substrate in order to continue development. So this shows the importance of the female to select the proper spawning site. So there are these interstitial spaces between the substrate particles to provide cover for the fish to continue maturing. These three eggs here, they probably are viable eggs, but they somehow got exposed to sand grains. After the eggs touch water, they become sticky. So these eggs are completely covered with sand grains and are probably going to continue to roll downstream in areas potentially that were not selected by the female. So when these hatch out, if they even can hatch out of this, they may not have substrate where they can hide and continue their development. Would be easy prey targets. So after hatching, this is a few days after, some development has occurred. You can see the eyes starting to form, the barbel down here starting to form, and then about 2 to 3 weeks after the eggs were laid, you have what's a larval sturgeon, which looks just like a small sturgeon. It's got the developed mouth, barbels, eyes, pectoral and pelvic fins, and is capable of some limited swimming, but is still dependent upon water flow to disperse them downstream from the spawning site. Just a quick overview of critical characteristics. For sturgeons in general, they're ancient, up to 200 million years old. They can live over 100 years, some species. They exhibit diadromy, patadromy, and amphidromy strategies. They can exhibit extremely long migrations. And this example is from the Danube River, where they migrate 1,500 kilometers within the river system itself, aside from what kind of movements they were making at sea. A wide range of adult sizes, they're late to mature, and they spawn infrequently, unlike some species that can spawn every year. They generally have discrete spawning sites, so there's a limited ability for them to use alternate sites, but they are colonists, They do have the ability to break into new areas. During the last glaciation, the glaciers extended all the way down to Long Island Sound. So all of the New England rivers had to have been colonized by, coastal wandering short-nosed sturgeon. And in this country, the short-nosed sturgeon have both federal and state protection status, as do many of the other sturgeon species. So on to the Connecticut River. Now this is the understood range of short-nosed sturgeon, roughly 197 kilometers from the river mouth at Long Island Sound to the Turner's Falls Dam. And it is believed that the Turner's Falls Dam was a plunging waterfall, and sturgeons have not been identified as having the ability to jump like a salmon to overcome obstacles like waterfalls. And I'm also going to spend a little bit of time talking about the Holyoke Dam. But for this slide, the interesting element is that this population of shortnose has a long history of study efforts. So here's the Conte lab, the University of Massachusetts, Holyoke Gas and Electric and associated consultants, as well as the Connecticut DEEP, have all been conducting various investigations on sturgeons, in some cases for decades. So it is a very well-studied species, yet there are still issues that we think could benefit from additional investigation. So we and others have published a number of documents detailing their critical life history. And in this case, I'm going to share with you their annual movement patterns. So the summer fall, which is actually late spring to fall, we call the foraging area. And really it can occur anywhere within this range. They usually disperse because they're in competition for food. And there are instances where there seem to be more aggregation of spawning, foraging activity. When the winter months arrive, they form wintering aggregations. And these blue circles are 6 of the known documented areas where sturgeons aggregate during the winter. And usually they return to these aggregation sites year after year. So in the spring, when fish are, those elements of the population that are bent on spawning, they exhibit 2 different migration strategies. One, we'll come back up here, is a 2-step pre-spawning migration strategy where fish, for instance, from the lower river would move upstream, and Holyoke Dam sits over, it's more over Hadley Falls, which is really more like Hadley Rapids. But it was an obstacle that they had to overcome in order to go farther upstream. So some of them would move up during the foraging season and spend the winter here, which is step 1. And then step 2, they'd make the final push to the spawning site here at Montague as part of the second step. But we've also seen fish, even from the lower river, make a one-step movement from the wintering site all the way up to the spawning site. So a number of strategies that we've observed fish make. And the Montague and Holyoke sites are the 2 places where evidence of short-nosed sturgeon spawning has been documented. And the evidence at Holyoke so far has been a minimally successful spawning efforts. And our evidence indicates a large portion of successful spawning occurs up here at the Montague site. But there is still ongoing investigation in attempts to identify additional spawning sites, because there are rivers such as the Kennebec that have at least 3 known wintering spawning sites. Oh, I wanted to build this up a little bit more, but this is going back to wintering behavior. this is a photograph of, or it was taken from a video camera image, just hung over the side of the boat in the winter over a known aggregation area. And so this is basically how fish are forming these aggregations for about a 5-month period at this latitude. And their feeding stops, their movements generally stop. And so, you know, these are kind of vulnerable, these aggregations potentially. So identifying them is valuable for managers who have to decide on whether a company can put in a marina or do a dredging operation. So at this point, I'm going to step off onto 3 issues that are kind of current events for sturgeons in the Connecticut River. And we're going to start off with recent modifications at the Holyoke Dam. So when a fish that wants to move upstream encounters the Holyoke Dam, it has 2 choices. It can enter we call this the tail race canal, which is an artificial canal designed to accept the outflow from water used to generate electricity at the Holyoke turbines. Or they could swim upstream into this kind of riffly area and then across these set of pools that we'll call the spillway or apron pools and enter a second lift called the spillway lift. So there's the tail race lift, the spillway lift, and sturgeons have used both of them. But there's been some questions about how effective this lift is. So since 1975, when the spillway lift was constructed, we've had an average of about 3.5 sturgeons entering the elevator, ranging from 0 to 16 fish. And what the sturgeon has to do is identify flow coming from what we'll call the attraction flow. And this is water that, if the sturgeon enters this canal, it will be shuffled into the elevator where it can be lifted and released upstream. But in many cases, water spilling from this bascule gate kind of competes with the attraction water flow. So a fish coming up here may have a hard time finding this tiny bit of flow in the midst of this more powerful flow. So it was decided that some modifications were in order. And so this is standing on the dam crest, looking downstream from the bascule gate. This is a kind of a ramp that takes all of that spillage energy, literally throws it into the air and into this catch basin where all of that intense flowing turbulent water energy can dissipate. And it's not so obvious from here, but in the next slide, you can see the difference between the flow chaos for fish trying to find the attraction water canal and the new design, which kind of throws the water up and over the attraction water flow. And it's more visible at the ground level. So all this turbulent flow gets thrown into the catch basin, preserving this channel of attraction water flow. And as a result, this came online in 2016. The numbers of sturgeons being able to find their way into the Holyoke fish elevator increased dramatically. We still think that there are things to learn about this. In fact, there are ongoing studies to learn about what fish that are lifted do. I mean, are they showing up on the spawning grounds? There are studies to look at how well fish are passing downstream. And part of this effort was to also make modifications to downstream passage. So this is still an ongoing investigation. And we were very excited to see these 3 years following the opening of this new structure in 2019 and 2020, tailing off. I don't know what that signifies, but we'll be watching this for the next several years. So many of you may realize that we are in the midst of a very unique hydropower relicensing event. So this can be described as once in a career, or maybe even once in a lifetime, relicensing of 5 separate facilities on the Connecticut River. And so they're all displayed here. The 2 that we have determined are the most influential over sturgeon spawning at the Montague site are the Northfield Mountain and the Turner's Falls projects. And we're going to zoom in a little tighter on this. So here's the Northfield Mountain Reservoir, Turner's Falls Dam, and the other facility that impacts sturgeon spawning is the Cabot Station, which is not up for relicensing at this point. Just general basics. Flow coming down the Connecticut River can be diverted into, away from the natural riverbed and into this power canal, which then feeds turbines at the Cabot Station. And I think their maximum capacity is about 14,000 cubic feet per second. So we're going to zoom in a little tighter to the Cabot Station area. And during our investigations at the Conte lab, we have identified 2 sites where sturgeons were known to spawn, meaning tagged fish, tagged mature adults were tracked there and captured there, as well as the capturing of viable early life stages. What I want to point out with this figure is, you notice there's the same photograph 2 different times. Here there's plenty of water. Here there is much less water. So as the spring floods, the freshet dissipates, more and more of the water available gets diverted into the power canal. So if there's 20,000 CFS, there's water in excess of what the Cabot Station can use and gets dumped into the natural riverbed, in which case sturgeons can be attracted to this site up here at the Rock Dam. At some point during the sturgeon spawning window, which is mid-April to mid-June, to include, early arrivals and also the final out migration of the larva, the water level drops to below 14,000 CFS, and the majority of the water ends up servicing the turbines, meaning some of the parts of the spawning area get deprived of water. So we investigated this even a little farther. So here again is the spawning site in a different color. This is what we estimated to be the spawning site. And during these low water flows, which can occur during the critical window, this shoal right in the middle of the action gets exposed. So we actually walked out onto this shoal. Here's a view looking upstream at Cabot Station and downstream looking at the bridge. And it had been demonstrated by studies of lake sturgeon that changing water levels could actually de-water shoals where there could be sturgeon early life stages. So we did take the opportunity to go out and look, survey some of these dewatering events, and we did find stranded eggs. These are, there's about a dozen embryos in this photograph, and these are desiccated embryos, but these were all of the white sucker. And after numerous attempts at trying to locate sturgeon eggs, we never did find sturgeon eggs on this shoal. However, even before these hydropower relicensing negotiations are concluded and final decisions are made, the utility has taken steps to avoid these dewatering events. So they have preemptively taken steps to preserve the integrity of the sturgeon spawning site right out in front of Cabot Station. So I'm going to talk a little bit about range extension. Now I'm going to zoom us back out, and here's the Turner's Falls Dam, and up here's the Vernon Dam. And again, this was thought to be the natural upriver extension of their range, because sturgeons cannot leap over a waterfall like a salmon can. Lo and behold, in 2017, an angler caught this sturgeon just a few kilometers downstream from the Vernon Dam. And my eye, after seeing thousands of sturgeons, this to me looks like a young fish, adult size, but still young. There's no scars, there's no deterioration. Fin margins are nice and tight. The scutes are nice and developed. So it looks to me like it's a young adult. And that tells me makes me ask, is there spawning happening up here somewhere? But I got to thinking, you know, I could count on 3 fingers a number of anecdotal reports of sturgeons upstream of Turner's Falls in my 20 plus years at the Conte lab. You know, if they were up there, we would have heard about it, right? And how would sturgeons even get up into that area? Well, between 1798 and 1856, there was a commercial barge canal and lock system, allowing commercial barges to be pulled upstream by beasts of burden to get over the Great Falls, which is the present site of the Turner's Falls Dam. So the blue portion is no longer used, and the yellow portion has been incorporated into the now existing canal. And these are just a few shots of the remnants of that old barge canal system you can find around town. So there was but you would ask, well, how do you really know that they would use something like that? To get up through 10 locks seems unlikely. Well, this is the C&D Canal, joining the Chesapeake Bay here in Maryland to the Delaware River in Delaware. And there are published scientific papers documenting sturgeon traversing this canal. And in 2012, a pedestrian took this photograph of a sturgeon in a lock associated with the Charles River Dam in Boston. So we know they'll enter locks. So it's not inconceivable to imagine that over the 60-year period of this barge canal system in operation, that a number of sturgeons could have relocated themselves upstream. And so just a final a note on this. As I said, you know, I've come across little or no evidence that there were sturgeons up there. And until 2017, you know, thought that I mean, you always have to take anecdotal sightings with a grain of salt. But once the sturgeon was photographed, we received funding to go conduct an investigation. And now that I'm up in this area, setting gill nets, encountering the local people, there actually are quite a number of sturgeon stories to be had. This figure shows the 3 dams, Turner's Falls, Vernon, and Bellows Falls Dam. And the blue stars indicate all of the anecdotal sturgeon sightings that I've collected in the few years that we've been, you know, mucking about up there. And again, you have to take anecdotal sightings with a grain of salt, but several of these sightings I have a great deal of confidence in. So it's not just in my mind, not just a lone odd event of the angler catching the sturgeon. It seems like there may be more than just a few up there. And the question to me and to others would be, you know, is there a reproducing population up there? And after talking to the people responsible for managing the short-nosed sturgeon, the Endangered Species Act provides protection for sturgeons, even if they are artificially moved into a location that was not selected for them by nature. So those are the 3 major issues that, you know, I and others are currently involved in investigating. And, you know, we have no shortage of ideas about what other studies could be done. but funding can be challenging, and we are at the Conte lab, very limited in our personnel resources. But I wanted to end with what I consider one of the most enjoyable elements of being out in the field and conducting these investigations, and that's outreaching to the public and partnerships. This photograph represents a collaboration of the National Marine Fisheries Service, and so Max here is representing NOAA's interest in. There's a proposal to conduct a pretty large-scale dredging operation to create a commercial shipping turning basin. So they were curious and provided us with equipment to survey that site. Our sturgeons going there and hanging out. Sturgeon an issue for that project. And on the right is Ian, who is an employee of the Portsmouth Naval Shipyard. And they build ships there and repair ships. So they have a number of large dry docks in which sturgeons could be entrained. So they provided some support to the project in order to investigate the vulnerability of sturgeons interacting with their facility. This program was started a few years ago by the National Marine Fisheries Service, and it is a public outreach action, and it's called the Scutes Program, and I have to read this each time. Scutes standing for Students Collaborating to Undertake Tracking Efforts for Sturgeons. So it works, but this is a curriculum designed for elementary, middle, and high school students. And participating members of this program can actually come to your school and present this curriculum, or teachers can go to this website and download materials and conduct their own sturgeon lesson plans. And a link for this should be available through the CRC. I'm also participate in a carcass recovery network. If you're an investigator and you desperately need to look inside a sturgeon to learn something about how to tag it or its physiology or age structures, they're mostly all protected, so it's really hard to come by specimens. But occasionally nature throws one up on the beach, and I'm kind of on call to go respond to these occurrences, to either gather the carcass itself or tissue samples or just general information. So the fish on top showed up on a beach in Cape Cod, and we hiked out about a mile in one of the coldest Februaries I can remember. The larger fish down below, which is measured about 6 feet, washed up in the Merrimack with a hideous gash in the side of its head, probably a prop strike from a larger vessel. And prop strikes is something that managers of these species are trying to get an idea of, how often does that happen? But unlike a road-killed animal, fish that get their heads cut off rarely make it to the beach. So this was a rare opportunity for us to go down and collect this fish. And on the same day, I had asked for the help of a volunteer who works for a local boat building museum in Amesbury, the Lowell's Boat Shop. And he said, well, yeah, listen, we've got a group of kids taking a class out on the river. What say we throw this big fish in a boat and run upstream and show it to them? So we got to do a spontaneous sturgeon demonstration of a pretty remarkable animal. Over here on the top right, oh bugger! Well, there goes my climax. But I'm showing the sturgeon I collected on the Cape Cod beach to the National Park Service staff. Down in the lower left is we participated in a mentorship program in USGS, and Isaac was with us for the summer. He was a junior in high school at the time. And then this is the 2018 River Festival held in Brattleboro by the CRC. And behind me is our Sturgeon Road Show. Everything can fit inside the pickup truck, the sunscreen, the posters, and if you look carefully, there's a live tank with a live sturgeon inside of that. So people are able to, hear my dog and pony show about sturgeons in the Connecticut and also have a good look at them. So I did not come up with this. I saw this at another sturgeon meeting, but at the time it seemed to hit the nail right on the head. So I thank you for your participation and interest, and I'll turn it back to Stacy.
Wonderful. Micah, thank you so much. That was so fascinating. And I'm glad you mentioned that you had come to one of our events and brought the live sturgeon. That was fabulous to be able to see it in live, but I feel like I learned so much from this presentation of getting the big picture. So kudos to you for sharing all of this backstory with us. It's really wonderful. I also want to mention that on the line now also with us today is Andrea Donlin and Kathy Erfer. They're our river stewards in Massachusetts and New Hampshire, Vermont, respectively. And they've been doing a lot of collaboration with Micah in different ways and are very active in the hydro relicensing efforts. So may also be able to speak to some questions if those questions come up. So just reminding folks to ask your questions in the chat box. And Micah, you can unshare your screen and then we can go into gallery view and see everybody who's here. And thanks again to everyone for being here. So I have a question here for Micah. Can you talk about sturgeon research in the ocean?
There certainly is a lot to be learned about sturgeons in the ocean. The Atlantic sturgeon, for instance, spends most of its life in the ocean. Excuse me. The challenge with that is the ocean's pretty big. And generally you need ocean-going vessels in order to, interact with the species while they're in the ocean. Researchers in the Gulf of Maine have tagged hundreds of Atlantic sturgeons that have been captured in rivers or nearshore environments. But the range on our data loggers is really pretty small when you compare it to the vastness of the ocean. So there's an ongoing effort of trying to collaborate with other organizations such as the NOAA weather buoys that are positioned offshore. And, can we hang receivers off of those? There was a program that requested that lobstermen put receivers on their lobster pots. So there is this growing effort to try and gather, get a bigger monitoring presence out in the coastal zone to try and gather. I mean, there's literally hundreds and hundreds of sturgeons swimming around with these acoustic tags on it. And I wanted to bring this up to the larger group. I just saw an email or a news article about a new wind turbine farm that has been, I guess, approved for, and it's somewhere off of New England. I'm sorry, I don't remember where, but can we lobby? They haven't started construction yet. Can we lobby for there to be a place for acoustic receivers to be put on all these wind turbine towers that may be erected? But getting back to the question, a lot of the information we have about Atlantic sturgeon is just what they're doing in the rivers. And again, it is much more difficult to gather information from fish in the ocean. Now, there certainly are noteworthy attempts. There's been an ongoing study of Atlantic sturgeon in the Delaware Bay. That's still kind of an enclosed habitat, but they've made some great inferences from those data. I know some of the researchers in the Gulf of Maine have used robotics, gliders and ROVs to kind of swim a receiver around. You can program them to conduct a search pattern. So, but nevertheless, identifying where those critical habitats are for Atlantic sturgeon is very difficult. Do they winter like short-nosed? Do they form these dense, vulnerable aggregations? We really don't know so much about that.
Okay, thank you. Here's a question from Ralph. From what I understood, there currently is no fish elevator on the dam separating the upriver newly discovered Connecticut River population. Have there been proposals to add a fish elevator to the separating dam for the endangered short-nosed sturgeon?
So I'm not sure I know what separating dam is being referred to. Is that the Turner's Falls Dam or the Vernon Dam, or?
Maybe Ross can.
I know there are fishways on both of those dams.
Turner's Falls, he says.
Okay, so there is a weir type fishway on Turner's Falls Dam. It can be difficult enough to get very expensive fish passage structures built, even if you know it's a good and will likely be a productive thing to do. Until we really verify that there is sturgeons upstream and that there is spawning occurring upstream, even then, the argument can be made that, well, nature hasn't selected sturgeons to be spawning upstream, if indeed they entered through a man-made canal. So there would have to be I would think, a very intensive discussion about, even if we find that there's a population up there, do we support it? I mean, I don't know the answer to that, but right now that is premature. Constructing fish passage like that.
Here's a two-part question. So how rare is it to see a sturgeon when paddling on the river south of Turner's Falls? And are the numbers of sturgeon increasing, decreasing, or holding their own on the river?
When people ask me that question, I advise them to launch their boat at the 116 bridge in Sunderland. And there's a nice, long, gently curving, 5 or 6 kilometer long reach that's pretty shallow in the summer. And if you're very quiet about it, have some polarized sunglasses on, you'll probably see sturgeons off the bow of your canoe. My experience has been that they are pretty skittish around large shadows approaching them. So just being quiet, that'd be my recommendation. Or you can go over to the Merrimack River near the American Yacht Club during May and June, and on a nice calm day, most of the fish you see jumping there will be sturgeons. Regarding the second part, I'm sorry, I forget the second part. Could you repeat?
Sure. Are the numbers increasing, decreasing, or holding their own on the Connecticut?
We did a population estimate on the group of fish between Holyoke and Turner's Falls Dam 20 years ago, and came away with about an estimate of 400 adults. But the state of Connecticut has been much more diligent in their population estimates of sturgeons in the lower portion of the river, and they're estimating, please don't quote me on this number, but 5,000 to 7,000 individuals, and charting an increasing trend.
Great. Hillary has a question that I hope was answered, was about, is there a safe way for people to look at the sturgeons? And I think you mentioned go quietly, unobtrusively. Is there anything more you'd like to say to that?
Well, they do. I don't know if Boston Aquarium has any. You may shop around for aquariums that might have some on display. You can snorkel, provided you are very reserved in your movements. I know that has been successful. We've scuba dove next to sturgeons, and they really seem to dislike that. Now, could it be the bubbles? Could it be that we were just inattentive? But that, other than catching one, which I do not recommend, you kind of have to be lucky to see them.
Great. Just note that Angie mentions the Springfield Museums in Massachusetts has a nice display tank of live sturgeon and other fish and animals in the basement of their science museum.
Well, there you have it.
There we go. So we have a couple more minutes, so we'll try and get in a few more questions from Rebecca. Oh, sorry, that's a comment. Are there fish hatcheries for sturgeon? Barbara would like to know.
There certainly are fish that are cultured. That brings up, what was at one time and still for the most part a serious conversation when discussing how do we restore populations of fish that have been extirpated, the term which means they used to inhabit an area and they no longer inhabit that area. They're not extinct, but they just don't occur in this area anymore. And so one technique is to, grab sturgeon A and sturgeon B and make little sturgeons out of it and release them into the wild. And a competing philosophy about that is, you're putting essentially inferior specimens into the population. Nature did not select which sturgeons spawn together, nor did it select which embryos survive. So you're introducing genetically and behaviorally inferior fish that will reproduce with the naturally created fish. And the theory is that they would then water down the evolutionary strength of the wild population. So interestingly, I participate in the Connecticut River Atlantic Salmon Commission, and we had this discussion a few years ago about, should we consider, or what do we need to know to consider a reintroduction of Atlantic sturgeon in the Connecticut River, which they used to come here and, gravid females were captured. So they used to spawn here. And, so we called all of the knowledgeable legislative bodies to say, what do we have to do to even have this conversation? And not too long after that, evidence of Atlantic sturgeon spawning in the Connecticut River was uncovered by Connecticut DEEP, which basically demonstrates, if you clean up the water and reduce as many of these, population reducing stressors, sturgeons are colonists, and it's within their behavior to, repopulate areas where all the environmental conditions are right. So by doing nothing, these Atlantic sturgeons were able to find their way into the Connecticut River and start to reproduce. The trouble with Atlantic sturgeons is it can be 10, 15, 20 years before a sturgeon spawns for the first time. So if you're looking for a quick return, sturgeons aren't going to give that to you. So right now, the prevailing philosophy, at least for the Connecticut River, and I'm guessing for many other sturgeon rivers, is just clean up the river, remove the obstacles, and wait. And hopefully that, sturgeons will find their way back. But again, it is a long-term view of population recovery.
Well, I think we're going to have to end it there. It's possible there's a few more questions. Maybe if Micah has 5 more minutes to hang on after, but I know that in the interest of time, some people will have to log off. So I just wanted to first say thank you to Micah for sharing your time and your expertise.
My pleasure.
And to everyone for joining us today. You'll receive an email later today with all of the links that were Micah mentioned, all the resources, and how you can get access to the recording and our future episodes. And Kathy has mentioned there's a upcoming Hydropower coffee hour conversation on June 8th, where you can learn more about the fish ladders that are going under relicensing now. And also Micah's contact will be posted in the email. So if we didn't get to one of your questions, please feel free to reach out, and to any one of us here at CRC. So thank you again, everybody. And I don't know, Micah, we'll say goodbye for the purposes of ending the meeting officially. And Micah, if you have 5 more minutes or anybody wants to stay on.
Yes, absolutely.
Okay, but I invite others, if you need to go, to please do, and we'll give it 5 more minutes for a few more questions. There is someone, if Terry is still here, wanted to know if there's any presence of sturgeon in the Green River, or are there too many obstacles to passage there?
So when we're out there conducting an investigation, You know, we typically have, you know, limited resources. So we let the tagged fish kind of tell us where to go. And we have tracked fish as far up the Deerfield River as to the mouth of the Green River. Now, the Green River is small enough that I can't really get in my boat and go upstream and track. So I would have had to adopt some sort of, you know, remote detection. But there never was enough, you know, critical evidence to lead me to decide to invest resources in answering that question. For the most part, sturgeons are a main stem river body organism, but they do come into tributaries. They've been captured in the Chicopee River. There's been sightings of them in the West River up in Brattleboro, and we actually thought that the spawning may be occurring in the Deerfield River when we first started this effort many years ago. But it turns out that it seemed like they were using the tributaries more like a refuge, as they're, you know, migrating upstream during the tail ends of the freshet. They could pop into the mouth of the Deerfield River and get away from all that turbulent, cold Connecticut River water and wait till conditions improve. So there has been no effort to survey the Green River, nor has there been any evidence to suggest that it needs to be.
Okay, great. Terry answered, thanks for answering. I thought that might be the case. We saw a good presence of lamprey in the near Green River Park. So I wanted to ask. I don't see any other questions though, but I do see that Sean added something where you were asking about on the wind farms. There are or will be acoustic receivers for sturgeon in many of the research buoys in the wind farm lease areas.
Fabulous.
So I don't know, Sean,
I
if you're still here.
Couldn't imagine that other people haven't recognized that and acted upon it. But I thought this would be a place to, you know, throw that out there, and I'm delighted to see that, you know, there is movement on that front. So bravo.
Great. So any last questions before we close out, or comments?
I had a question if nobody else does, but I'll let others chime in.
I think you're good to go, Andrea.
Okay. Hi, Micah.
Hi.
I think there's a management plan or recovery plan for short-nosed sturgeon, right? And it's sort of old. Is it like 1990?
You know, we did we did a status review not too long ago, but as far as an actual recovery plan, I think you're probably right. 1988 pops into my head, but there may be one more recent than that as well.
I was just curious if you had any insider knowledge about how often those come up, or whether that one is going to be updated at some point relatively soon.
I know status reviews for, you know, at least nymphs managed species are supposed to be conducted every 5 years, but it's probably more like every 5 to 10 years. And again, they just did do one, and they are time consuming and involved events, and they did reviews for, you know, a number of sturgeon species back then, all of which have been published. So I don't know if there is if a study plan is a regularly scheduled event, or does there have to be some sort of critical mass before one is called for?
I see. Okay.
But I certainly can look into that for you if you like. I think nymphs would probably be the contact for that.
Yeah. Well, if the status update is a public document that's online somewhere, maybe we could link to it in our reply to all the participants.
Mike, I have a question. Given the number of anecdotal sightings or captures, like north of the Mass border, would you want to have a more dedicated kind of community outreach effort for anglers if they do catch a sturgeon, to notify you or try to document it?
When this whole story first broke, I went to the the visitor center there in Turner's Falls, the Fish and Wildlife Service, and asked if I could put down a little information blurb and a logbook. So if people visiting the center had information like that, they could enter it in there. And I never really saw that through, but just like the example of, you know, local knowledge has a tendency to stay local, that could really help narrow this search, because we're talking about, you know, 80 kilometers of river, and that's a pretty large chunk of river to poke and hope. You know, I can guess where they might be, but I've set nets in the midst of tagged fish before and came away with nothing. So there is an element of luck involved that, you know, anything that can help minimize that would be helpful. So perhaps we can converse about places that you know, invite the public in for various reasons, you know, ecology interested people, and I can get back to work on crafting a response, you know, set of information in a logbook that could maybe see that through. The other thing that I haven't really investigated in, and I plan to, is, you know, I've kind of avoided Facebook, but I understand there are, you know, chats that are occurring specifically about sturgeons in specific areas. I know there's one on the Merrimack, and I need to be able to tune into that, because they can be a wealth of information.
Yeah, we'd be glad. I'd be glad to help with that.
Okay.
We've been doing, and Aliki's on the call here too, who does some of our community science work. We've started doing, you know, stream walks to look for sea lamprey nests up our way in Vermont, New Hampshire, and you know, often are communicating with the angling community along the river. And so I think just being able to put the word out, like if you happen to catch a sturgeon, let us know, or let Micah know, you know, would be a relatively easy thing to do. Yeah.
Because, you know, even without having a hired hand, I do have access to a core of volunteers, and still can potentially respond to, you know, a hot lead. You know, I saw a sturgeon here yesterday, and I've done that in the West River, you know, on 2 occasions, and one of them was the Brattleboro River Festival. A gentleman indicated that he'd seen some, you know, up the West River, and I was there shortly thereafter with nets. And there was a scuba diver sighted a sturgeon by the old bridge pilings in Northfield. And we were right on that with nets, but, you know, it could be one sturgeon and it's long gone. So you kind of have to put in the time of putting out a lot of effort. It has been my experience, unless you have a real, you know, burgeoning hot lead that, you know, you can go exploit.
Great. Well, thank you, Micah. Let's continue to, you know, stay in touch and see how our organizations can work together. Really appreciate everything you've given to us today. Thanks so much.
Again, my pleasure.
Yeah. Okay. So we will close out here. Thanks, everybody.
Thank
See you
you.
next time.